Similarly to the GMOs era, previously to the huge commercializations, there are 3 or 4 mainly topics regarding to that. What's happening with GMO? They decide to do a kind of agronomic studies or check if it's the new variety will become a weed or not, or something like that. So they carry on some research studies that will not indicate any risk or something like that. This happens with some articles, some studies, that they do some checking or some traceability, in the sense that we're looking for with one technique that just look for similar sequence in DNA. So then maybe are not changed like the target site. So say is a kind of partial tentative to look in all genome. So they also don't find too many side effects of targets. The second issue is that most of the studies try to optimize the protocol to develop a modified organism. So not in the sense that looking for effects on this of the technology. The third issue is that the people that were behind, so the labs that were behind the GMO research or development, now wants to be in place or to provide some development, some products. So there are many small industries and labs across European countries that they are using these gene edition technologies to develop products. So this didn't happen with the GMO because only big companies try to develop. And the last issue is that we don't know or we know very little, in other words. We do have one study that was published maybe 5 or 6 years ago in mice. That was a huge number of targets, sites that were reached by the same protocol that reached the target site. And also this is a classic study that showed several effects on the organisms. However, in plants we don't have too many studies. So, and we don't know how many sites are changed across the genome of the one plant. And also we need to study effects on the physiology, on the biochemistry, on the genetics that we didn't carry out this kind of studies with GMO in the sense to looking for the entire genome. So contrary to GMOs that can insert one or few transgene or DNA fragments, this technology can reach many sites in the genome. And so we don't have even the notion of the expertise to expect how many sites will be affected with one gene edition delivery. So I guess it's too early to say that we have effects on phenotype or in the genotype and will be the magnitude of the effects in each site. So we don't have this kind of experience yet. In my opinion, it's too early to say something. But since it's a modification, we can also take in consideration that if the DNA sequence that is inside one plant or one animal is there for millions of years together, they are the rate of evolution is there, so the sequences are together for a long time. So if we have many modifications in one genome, probably you can expect a kind of provocation in order to redefine the evolution of this organism. So we don't know about if the adaptation will be similar, or if the adaptation of this new organism will require a new environment, or if a particular site that was changed will be provoke a very huge effect, phenotype effect or not. So we don't have this kind of experience.